Essay: How energy is transferred within and between organisms. Energy is needed by all living organisms and provided by respiration. Respiration releases energy needed for the movement of molecules across membranes around the organism and movement of the whole organism in some cases. The ultimate source of energy is from the sun in the form of sunlight energy. This is captured by the plants in a process called photosynthesis, and this generates chemical energy in the form of biomass in molecules such as starch and protein and cellulose. The light energy is captured by molecules of chlorophyll which trap the sunlight energy, and this causes electrons to become excited and pass along the electron transport chain in the thylakoid membrane. As the energy in the electrons is passed along the electron transport chain, it causes hydrogen ions from the photolysis of water to be moved into the thylakoids. When there is a proton motive force, it allows the enzyme ATP synthase to generate ATP from ADP and inorganic phosphate, as the hydrogen ions move down the concentration gradient from the thylakoid into the stroma through the ATP synthase enzyme. In non-cyclic photophosphorylation, the electrons are then excited again in photosystem 1, and this allows the electron energy to be transferred to another enzyme for the production of reduced NADP. The reduced NADP and ATP are then transferred to the Calvin cycle in the stroma, and this is where carbon dioxide from the atmosphere is converted into carbohydrate in the form of hexose sugars. The hexose sugars are then converted into other molecules and make up the biomass of the plant. Some of the sugars are used up in respiration as part of the production of ATP from glucose. The energy released as part of respiration cannot be passed on to other organisms, and so the equation that summarises the energy generated in plants and available to the next trophic level is gross primary production equals net primary production plus respiration. The energy from plants in their dry biomass can be ascertained by drying the plant, heating in an oven at 100 degrees centigrade. Any higher may cause combustion, and so any loss of mass may be due to more than just the evaporation of water, but actually the combustion of molecules. The process is repeated, weighing and heating, until the mass remains the same. The final mass ascertained is the dry biomass. This can then be burned in a bomb calorimeter, and the energy supplied can be calculated using the equation which takes into consideration the heating of a set volume of water. Joules of energy equals the constant 4.2 times the rising temperature in degrees centigrade times the mass in grams. Once the amount of energy found in a particular plant has been determined, the net primary productivity can be calculated, and this is the maximum amount of energy that can be passed on to the next trophic level. However, if not all parts of the plant are eaten by the organism in the next trophic level, for example, cows do not tend to eat the roots of grass and humans do not eat the leaves of rhubarb, then not all of the energy available will be passed to the next trophic level. Energy is lost through the ecosystem as part of energy transfers as well through respiration by the organism in the next trophic level. There is energy losses due to the molting, loss of hair and other proteins, for example in the horns and antlers, that can be lost in the environment. The loss of energy from the consumers in a food chain can be summarised as net productivity equals intake minus, in brackets, fecal matter and respiration, where NP is the net productivity, that is the energy that is passed to the next trophic level. We use I for intake, which is all the energy that is consumed by the organisms in terms of biomass, and then F stands for feces and urine losses, and R stands for losses due to respiration, remembering that respiration involves lots of exothermic reactions producing heat energy, which can then not be trapped again and is lost to the environment, and so lost from the food chain. The efficiency of transfer also relies on the food taken in being completely digested. This is not always possible. For example, some organisms do not fully digest cellulose. This chemical energy is lost as part of the feces and is not available to the next trophic level. However, fecal matter and urine contain compounds that can be broken down and hydrolyzed and recycled by the saprobionts that are found in the soil. These bacteria can break down the compounds found in feces and also in the skin and hair of organisms, as well as the molecules in dead organisms, and break it down into the basic components of the nutrients, such as nitrates and carbon. And these can then be taken up by the plants and so re-enter the food chain through the organisms. This is because when the bacteria start to respire, they release carbon dioxide into the atmosphere, and so plants can absorb the carbon dioxide from the atmosphere through their leaves and the nitrates that are produced by the bacteria in the soil can be taken up by the plant along with water. Farmers may use their knowledge of the provision of nutrients to the soil to improve yield. Gardeners utilise the fungus mycorrhizae to improve yield in plants, since this fungus forms a symbiotic relationship, allowing plants to take up more nutrients and water so that plants can have a higher surface area in their root system, and this improves growth rate and yield. At the next trophic level, farmers need to limit losses so that net productivity can be maximised. This can be done by reducing movement of animals, providing warm places to live, to limit respiration, to maintain body temperature, by providing food that needs minimal digestion and food that can be completely assimilated into the organism's own biomass. Also, treatment for pests at both the level of producers and animals is important for the conservation of energy. Herbicides can destroy plants such as weeds, and the loss of these plants can help to reduce the competition for light, water and sunlight, which would limit the growth of the plant. Insecticides can destroy insect pests that destroy crop plants and limit yield. Insects that carry disease are also destroyed to reduce infections in animals. Infected and diseases animals have a lower yield of biomass to pass on to the next trophic level. So to help improve their net productivity, farmers can give animals antibiotics to reduce infection levels, insecticides to destroy disease-carrying pests, and vaccinations to help prevent infection. These chemicals and injections all cost money, but they also do help to improve the net productivity. Once an organism consumes the net productivity from the previous trophic level, the food must be digested by hydrolyzing enzymes. Some organisms have symbiotic relationships with bacteria. Without the bacteria, the organism would be unable to digest and therefore limit the useful biomass that can be assimilated into the organism's own biomass. For example, cows carry bacteria that have the enzyme cellulase, which is able to break down cellulose in plants and allows more carbohydrates to be released. This carbohydrate is used by the animal as a source of energy for respiration. Respiration is used to generate ATP, and ATP is needed for processes such as active transport, which are needed to complete absorption of glucose and amino acids from the gut following digestion. ATP generated as part of oxidative phosphorylation is done inside cells that require ATP directly, as ATP is not circulated within the organism. But glucose can be circulated within the organism, and this is done in the blood. The level of glucose in the blood at any one time is closely regulated by homeostasis and the two hormones insulin and glucagon. These hormones ensure that the levels of glucose never go too high. Insulin release causes more glucose channels to take up the glucose into the muscle cells for use in respiration, which produces ATP, and this is then used in muscle contraction. Glucagon helps to break down more glycogen into glucose. Glycogenolysis, which releases more glucose from the glycogen stores in the liver, and this can be delivered to cells in the body. For example, during the formation of tissue fluid, glucose diffuses out of the capillary bed and into the tissue, where it is used in respiration to form ATP, and this is used in the cells for the processes such as cell division and exocytosis. ATP is also needed as part of translation, where it is needed to attach amino acids to the tRNA, and this is needed for the synthesis of new proteins which make up part of the biomass as part of the muscle tissue that is consumed and digested in the next trophic level. Respiration is exothermic reaction, and this will lead to loss of heat to the environment, so it's important for the farmer to limit the level of respiration in animals to help limit energy losses through heat loss. Respiration involves the stages of glycolysis, which converts glucose into more reactive substances, and eventually the production of pyruvate, alongside some ATP at the substrate level phosphorylation of ADP into ATP. The pyruvate enters the link reaction, and this is used to produce acetate, which is carried to the Krebs cycle in the mitochondrion, and this is where NADH and FADH are produced in the Krebs cycle. There are also more ATP generated by substrate-level phosphorylation, but in aerobic respiration, most ATP is synthesized during oxidative phosphorylation. The oxidative phosphorylation uses energy from chemiosmosis of hydrogen ions into the intermembrane space, and this proton gradient drives the ATP synthase enzyme to produce ATP as the hydrogen ions pass back through. The ATP generated in the muscle mitochondria needs to be transferred to the muscle fibers, actin and myosin, where it allows muscle contraction by facilitating the formation of cross-bridge following nervous stimulation. ATP cannot move directly there, so is involved in a reaction with creatine. Creatine reacts with ATP and forms creatine phosphate. This travels to the myosin, and this is where the ADP is then converted back to ATP by removing the phosphate from the creatine phosphate. The creatine then returns to the mitochondria and is recycled into creatine phosphate. The final transfer of ATP within the organism involves nervous impulses. In order to transmit a nervous impulse to a muscle cell to cause contraction, and the hydrolysis of ATP, an action potential must be generated. An action potential can only be generated in a neuron that has been depolarized and then repolarized. This requires ATP, which is needed to power the sodium-potassium pump, which moves 3 sodium ions out of the neuron and then 2 potassium ions into the neuron. This mismatch of ions causes the resting membrane potential to be held at a negative minus 70 millivolts. To summarise, the transfer of energy between organisms involves ingestion of organisms along the food chain, and the transfer of energy within organisms involves digestion and absorption, and glucose is then transferred to the blood in different cells, and within cells, creatine phosphate can transfer energy in a muscle cell from one part to another, but ultimately it is ATP that provides and acts as a source of energy initially.